Yeah. F. Hannah, what is your name? F. Hannah? Hello? Shall we start, ladies?
Yes, doctor, we can start.
Okay. I think you already had a practical session on the brain or the cerebral in particular. Have you? Did you do it with Amina?
Yes, doctor.
Yes, doctor, we do.
So you already know about the cavities in the brain. You do? And you already covered the meninges. I think you have. So who can brief us on these two issues very briefly? Anything about the meninges and anything about the ventricles or the spaces? Because the ventricles are part of the spaces, there are additional spaces. So who can start telling us about, I will just put the PowerPoint, just.
Doctor, we covered the meninges, but we still didn't cover the ventricles.
Huh?
We covered the meninges, but we still didn't cover the ventricles.
You only studied the topography of the brain, the external features?
Doctor?
Yes?
Could I add something about the ventricles?
You can tell us something about the ventricles? Yes, please.
So you have 4 ventricles, 2 lateral, 1/3 and 1/4 ventricle. The lateral and the 3rd ventricles are connected by the intervertebral foramen of Monroe, and the 3rd and 4th ventricles are connected by the aqueduct of Sylvius.
Okay, excellent, excellent. Okay, so this is the briefing, the outline I wanted to hear. Thank you very much, Jumana. What about the, why am I talking about the meninges while we are discussing the ventricles? Why am I talking about that? Do we need to know any background information about the meninges? Why are they related to the ventricles? What is the relationship between the ventricles and the meninges? Tell me anything about the meninges, other than they are the Dura, Arachnoid, and Pia. What can you tell me about them? I will ask specific questions. You have a Dura, and this Dura is made up of 2 layers. One layer fixed to the bone, and another layer facing the brain, and of course the spinal cord. Now, do we have spaces between the 2 layers of the Dura?
Yes, the Arachnoid Space, the Sub Arachnoid Space.
No, I am now talking about the Dura. The Dura itself is made of 2 membranes, an outer membrane and an inner membrane. One of them is tough and fibrous. The other one is not quite thick and not quite fibrous. Between them, within, inside the Dura, we have spaces, very important spaces, very relevant to the lecture we are having today. What do you know about this? You already said you studied the meninges. And this is a very important piece of information.
Doctor, is it related to the cerebrospinal fluid?
Yes, and this is
Yeah.
why I'm talking about them. I'm talking about the meninges because the CSF is produced in the ventricles, but it circulates in relation to the meninges. So we need to know a little bit about the meninges so that when we discuss the cerebrospinal fluid circulation, we relate it with the structure of the meninges. So I started with the Dura, and I need just two pieces of information in relation to the Dura. The spaces around the Dura and the spaces in the Dura. The spaces around the Dura, external to the dural membrane, there is a space, and it is called extradural space or the epidural space. Some people differentiate between the two, but let us say they are the same thing. An epidural or an extradural, it is between the dura and bone. This is not quite related to the cerebrospinal fluid, but it's a very important space because it contains blood vessels. And when there is a skull fracture, we have bleeding in this epidural space. And this bleeding is called epidural bleeding, epidural hemorrhage. And when the blood clots in that space, it is called an epidural hematoma. And it is serious because it's a very tight space. And when there is a hematoma, it strips the dura a little bit, but it compresses the surface of the brain, leading to problems. Very well known epidural hematomas and paralysis and all sorts of lesions. Now, this is outside. Now, under or beneath the dura, between the dura and arachnoid, there is another space, and it is called subdural space because it is under the dura. This subdural space is also very important because it houses veins crossing from the subarachnoid space to join the third type of spaces I am looking for now within the dura. So the veins are crossing the subarachnoid space to join spaces within the dura. And these veins often bleed. When you have a traumatic accident and the movement of the skull and the brain, these veins are severed or cut, and they bleed in the subdural space. And because they are veins, the blood flows slowly, and the subdural space is an open space. The hematoma doesn't develop quickly. It takes time. This is why people come 2 to 3 weeks after an accident complaining of things. Then they discover that they have a subdural hematoma. So you need to read about that. So these are the spaces around the dura. The spaces in the dura, there are some spaces in the dura, and they are called dural sinuses, dural venous sinuses. Very important. They are mainly for the drainage of the blood in the brain circulation, the cerebral circulation. All the blood drains into these dural sinuses, and from the dural sinuses, it goes to the heart. So you need to know that there are spaces around the dura, there are spaces inside. How are they related? The cerebrospinal fluid also drains into these dural sinuses. So you need to know about the dura and the dural sinuses and these spaces. Now, between the arachnoid and the pia or pia meta is covering the surface of the brain, and there is a space between it and the arachnoid, very important space. It's called the subarachnoid space. It's very important because, one, it is the main site for the cerebrospinal fluid, and the cerebrospinal, actually the brain floats on the cerebrospinal fluid in the subarachnoid space. But in addition to that, it has got vesicles, and these vesicles often bleed, and the bleeding is called the subarachnoid bleeding. And this subarachnoid hemorrhage is very serious because it increases the intracranial pressure and it compresses the brain, and it's very fast because it is usually arterial bleeding or capillary, but it could be arterial bleeding. And arterial bleeding is fast, and it compresses the brain. It increases the intracranial pressure very fast, leading to major problem, even death. It is deadly. It is life-threatening. This subarachnoid hemorrhage. We need to know it because cerebrospinal fluid is also in that space, subarachnoid space. So you need to know about the meninges and the spaces within them and the spaces between them, because they are relevant to what we are going to talk about today. Now, having this, after finishing this introduction, very important introduction because it's very relevant to what we are talking about, we will be talking about in a little while. Bismillah ar-Rahman ar-Rahim again, and then we start talking about the ventricles. What we are going to cover is these cavities within the brain. Now, as your colleague Jumana mentioned, we have a cavity in the cerebrum, and actually two cavities in the cerebral hemispheres, one cavity between the cerebral hemispheres, and one cavity in the brain stem. So these cavities are known as the ventricles of the brain. There is a lateral one in each of the two cerebral hemispheres, and there is a third one between the cerebral hemispheres. There is an aqueduct of Sylvius found in the midbrain, connecting the third ventricle with the fourth ventricle, and the fourth ventricle is between the pons and medulla, roofed by the cerebellum, And then there is a septum between the lateral ventricles, and it's called the septum pellucidum. We will see it in one of the photos. Now, the lateral and third ventricle, as she correctly mentioned, are connected through an interventricular foramina, one on each side. While the lateral and fourth ventricles, yes, I already mentioned that they are connected through the aqueduct, but the lateral and fourth ventricles contain arachnoid plexus. And arachnoid plexus is very important. It's a vascular plexus, and this vascular plexus produces the cerebrospinal fluid. Physiologically speaking, they will tell you about the production of the CSF, but it is a production rather than secretion. So can anybody tell me what is the difference when I say it is secreted or it is produced? Just to keep you following, can you tell me about the difference when I say let me ask you the question, and then you start thinking about it, and maybe you will be able to answer. Is the CSF secreted? Is it secreted or rather produced? You forgot all your cell biology? Let me take the question one step back. What is secretion? Are you there? I have 26 attendants or attendees. Nobody of the 26 people in this lecture can answer any of these three questions. The most basic one is, what is secretion? Can anybody tell me about what is secretion? Are you awake? Are you there?
Yes, doctor, we are.
Yes, Jumana. Jumana, what is secretion?
Secretion is like flow in a way.
Flow is part of secretion because you need to synthesize something
Yeah,
inside
so
a
production
cell.
and
Yes, and then release. You synthesize and release. So is it right? The cell will have the machinery inside that actually synthesize something. Then through a process of exocytosis, it releases this product. Am I right? So it is synthesized inside the cell from basic elements, from raw material. We synthesize something. It could be a protein, it could be a hormone, it could be an enzyme, but we actually put elements together to synthesize something. Then through a process of vesicle formation, vesicle movement, and then exocytosis, we release. This is secretion. I can secrete mucus, I can secrete a fluid, a watery fluid, I can secrete sweat, I can secrete tears, I can secrete many things. So this is called secretion, when a cell actually synthesizes something and release it. But, halas, عرفنا what is secretion. Is the CSF secreted or it is rather produced? It is rather extracted. It is extracted. Lymph is not secreted. Lymph is extracted from the blood. So the material is already there in the blood. It filters, and then through a filtration process, something results, and this result is lymph, and this lymph is produced rather than secreted. Now, CSF is produced. CSF is an extraction of fluid from the blood, like lymph. Let us leave it at that. In the physiology lecture, we will go more in detail. And when I bring the illustrations and photos, I will explain this issue more. So there is something called the arachnoid plexus, and this arachnoid plexus is mainly in the lateral and the fourth ventricle. But you will find an extension of the arachnoid plexus of the lateral ventricle, an extension. Through the interventricular foramen, you can see some of the arachnoid plexus of the lateral ventricle residing in the third ventricle. So there is production of CSF in the lateral ventricle. It continues in the third ventricle because some of the arachnoid plexus of the lateral ventricle actually passes through the foramen, the interventricular foramen, and is found in the third ventricle. But the fourth ventricle has its own arachnoid plexus that produces more cerebrospinal fluid. Okay. Okay. Now, for the cerebrospinal fluid to move out of the fourth ventricle, the fourth ventricle has got three openings. One central one in the center at the back, and two side ones. The two side ones, the lateral ones, are the foramina of Luschka, and there is one in the center, one in the median plane, and it's called the foramen of Magindi. And these open into the subarachnoid space. So the fluid produced in these ventricles come to the fourth ventricle, and from the fourth ventricle it moves out into the subarachnoid space of the spinal cord and brain. So there are three openings in the fourth ventricle. These are the ventricles. This is the lateral ventricle, and this is the third, and this is the fourth ventricle. This is a side view. This is an anteroposterior view. We have a lateral ventricle on each side, on each cerebral hemisphere. There is a third ventricle between the two cerebral hemispheres. There is a fourth ventricle in the brain stem, and there is an opening between the lateral and third. This is called the foramen of Monroe, or the interventricular foramen. There is an aqueduct connecting the third ventricle with the fourth, and there are three openings. One central here is the foramen of Magindi, and one lateral on each side is the foramen of Luschka. Luschka describes these two foramina. And there is, of course, the aqueduct of Sylvius connecting the third and fourth ventricle. This is a larger view, and you can see them very clearly. This is the lateral, this is the third, this is the foramen of Monroe, the interventricular foramen, this is the aqueduct of Sylvius, this is the fourth ventricle. This is the openings, one on the side and one in the median plane. Lateral ventricle, third ventricle, Monroe, aqueduct of Sylvius, these are the interventricular foramina, third ventricle, aqueduct of Sylvius. This is, you can see here, are the foramina of Luschka on the sides of the fourth ventricle. Then I forgot to mention the cerebrospinal fluid continues into the central canal of the first, the spinal medulla, and then the spinal cord. And even the cerebrospinal fluid found around the spinal cord does not come from the central canal, it comes from the fourth ventricle through the foramen of Luschka and foramen of Monroe. Now, okay. Now, this is a very important section in which we can very clearly see this is the corpus callosum. It means through this opening, this is the entrance into the lateral ventricle, and this is the septum pellucidum between the two lateral ventricles. So we have one lateral ventricle on the right, one lateral ventricle on the left. Between them is a septum pellucidum. And by the way, if you are still awake, do we have connective tissue in the central nervous system? In the central nervous, because we have connective tissue in the meninges, but do we have connective tissue in the central nervous system? And while you are thinking about it, think about, do we have connective tissue in the peripheral nervous system? Anybody here? Shall we make this an assignment? Why am I asking about this? Because connective tissue, which functionally speaking, the tissue that connects or sits around functional units. If you have a functional unit or a functional basic unit, which is a cell, the functional units of the central nervous system are the neurons, and they need some supporting structures. We call them connective tissues. The connective tissues in the brain and spinal cord, in them are the glial cells. So we have the glial cells as connective tissue, and they, but what about this? What about this septum pellucidum? Septum pellucidum contains glial cells, but it also contains neurons and axons and all of that. It has got septal nuclei. There are nuclei in the septum. So it is not connected, it is not collagen fibers and elastic fibers and all of that. Although it's a septum, but it's a nervous tissue septum separating the two hemispheres, I mean separating the two ventricles in the two hemispheres, the two lateral ventricles. This is an important piece of information, the septum pellucidum. This is an open, of course, it is open because it's actually the space between the two cerebral hemispheres. And actually the walls, there are two pieces of information here, very important pieces about the third ventricle. The lateral walls of the third ventricle, because they are both lateral walls, are actually made of the diencephalon. So the thalamus and hypothalamus make the walls. And why is that important? If you have an issue in the lateral, in the third ventricle, these diencephalic elements will be affected. If there is a hemorrhage, if there is a hematoma, compression, if there is a tumor of some sort, then they will compress the diencephalon, leading to lesions in the thalamus, hypothalamus, and the other components. This is a piece of information number one. The other piece of information is related to the content You can see there, choroid plexus. It says choroid plexus of the third ventricle. It is in the third ventricle, but it's an extension of the choroid plexus of the lateral ventricle through this foramen, interventricular foramen. It's through the interventricular foramen of Monroe. We have an extension of the choroid plexus of the lateral ventricle, making the plexus in the third ventricle. Now, when you study the details of the ventricle itself, you will see that it has got some recesses. There is one related to infundibulum of the pituitary gland. It is called the infundibular recess. We have something above the optic chiasma, and it is called the supraoptic recess. It's an extension. And there we have the opening of the third ventricle into the cerebral aqueduct of Sylvius, opening into this space, which is the fourth ventricle, So this is a section depicting or showing you the lateral ventricle covered by the septum pellucidum, and this is the lateral wall of the third ventricle with recesses and openings and everything, two openings there and here. And then we have the arachnoid, choroid plexus. By the way, in the PowerPoint in the previous slide, it is written as the arachnoid plexus. Sorry for that. It is the choroid plexus. Here you can see the choroid plexus of the fourth ventricle. Yes. Okay. This is just the ventricles, just a reminder. But as you can see, this is the choroid plexus of the third ventricle. I want to add a lateral ventricle, and it extends through the foramen of Monroe into the third ventricle. So it is one choroid plexus. Now, in this diagram, I just wanted to mention a few things about the lateral ventricle. The first thing is that it has got horns. It has got an anterior horn, and it has got a posterior horn, and it goes an inferior horn. This is in the frontal lobe, this is in the parietal and occipital lobes, and this is in the temporal lobe. So this is important because we need to know that there is a floor, there is a roof, there are lateral walls, and these are bordered by brain tissue. And this brain tissue is in fact gray matter and white matter elements. You need to know what is on the related to the lateral wall of the anterior horn, what is related to the the medial wall of the anterior horn, what is related to the roof, what is related to the floor of each of these three horns. And then in the middle between the anterior horn and the posterior horn, there is a body and an atrium, a dilated area in the parietal lobe. And the dilated area is between the posterior horn and the inferior horn. This is the atrium, this is the body, this is the anterior horn, this is the posterior horn, and this is the inferior horn. This is the lateral. We saw the two recesses, one supraoptic and one suprachiasmatic. And then we have the aqueduct of Sylvius, and then we have the fourth ventricle. There is another choroid plexus of the fourth ventricle. Now, because of the time, I need to rush a little bit because I need to cover the fluid. Now, this is an important slide because I just wanted to mention very few things about the relationships of the lateral and third ventricles. Now, this is the lateral ventricle separated from the other lateral ventricle with this septum pellucidum. I already mentioned that it is made of brain tissue and brain connective tissue. So it is white matter, some glial cells, and some neuronal cell bodies making septal nuclei. This is an important piece of information. But related to the lateral wall, there are some important structures. There are the basal nuclei. We will study them in detail later on, but they border the lateral ventricle. The lateral ventricles are roofed with the septum pellucidum. So the septum pellucidum makes the roof of the lateral ventricles. This is point number one related to this section. Now, this is a higher magnification of a soft tissue, a very important view. You can now very clearly see the septum pellucidum. You can see the roof, which is the corpus callosum. You can see the interventricular foramen very clearly, and you can see the extension of the choroid plexus from the lateral ventricle into the third ventricle. Now, this is the third ventricle. Here you can very clearly see the supraoptic recess, the supra infundibular recess, not chiasmatic, sorry for that. This is the supra infundibular and the supra optic. Now, you can see this is the diencephalon. Later on, you will know the details. This is the thalamus, this large one, and the lower one there is the hypothalamus. This is the mammillary body, by the way, of the hypothalamus. So this is the hypothalamus. So the lateral wall of the third ventricle is actually made of the diencephalon. Now, this is the opening between the third ventricle into the aqueduct of Sylvius, and then the aqueduct of Sylvius opens into the fourth ventricle, and there is the fourth ventricle. This is a very important section. Now, very briefly, the Csf circulation, anatomically speaking, and I'm going to skip over a number of detailed description of the production of CSF, but I'm going to summarize the circulation in general. Circulation in general, where does it start and where does it end? We already mentioned that CSF is produced. And sorry, this is a repeated mistake. I will correct it before I upload the PowerPoint. It is the choroid plexus. So it is produced by the choroid plexuses in the lateral ventricles, passing through the interventricular foramen to the third. More is produced in the third. Then the CSF passes through the cerebral aqueduct to the fourth. More is produced in the fourth. Then some pass directly into the central canal of the medulla first, and then the spinal cord. Then through the throat foramina, which are the foramen of Luschka and the foramen of Magindi. Then through the now. Where do they go from they go to the subarachnoid space. So they surround the spinal cord and the brain in the subarachnoid space. Now, the subarachnoid space is lined with ependymal cells, ependyma. Ependymal cells is one of the connective tissue cells of the brain and the spinal cord, of course. Now, they line the cavity. And by the way, they look like endothelium of the heart, which is the endocardium, and the endothelium of the blood vessels. And it is very smooth because we need a very smooth surface for this fluid so that it doesn't clot like the endothelium of the heart and the blood vessels. Very smooth so that it doesn't clot. The fluid doesn't clot and the blood doesn't clot because it doesn't come into contact with a rough surface. So these ependymal cells make what is known as arachnoid villi, and these arachnoid villi actually project into dural sinuses. They project into, and this is why I started the lecture by revising the dural spaces. The spaces in the dura are called dural sinus. One very important dural sinus is called the superior sagittal sinus. It is the largest. So ependymal cells project through the wall of these sinuses. So they go through the arachnoid, they go through the internal layer of the dura, they go through the wall of the sinus, and then they project into the sinus, and they are called arachnoid villi. And this arachnoid villi go into the dural sinuses, but mainly the superior sagittal sinus. And they make collections. A collection of arachnoid villi is called an arachnoid granulation. And you will see them inside when you dissect the superior and open the superior sagittal sinus, you will see these arachnoid granulation. And they actually make impressions in on the surface. You can see them very clearly making impressions. Now, if the movement of the fluid through these arachnoid villi, if the fluid cannot go through the arachnoid villi, if this happens in the fetus or infant, it will lead to an enlargement of the brain and an enlargement of the head, and actually the skull doesn't actually fuse. The sutures of the skull remain open, and we have a condition known as hydrocephalus. Now, this is a brief description of the circulation. It is produced there in these choroid plexuses of the lateral and third and fourth ventricles. And then the fluid moves into the third through the aqueduct into the fourth, out of the fourth into the surrounding space. But if we go to the cellular level, actually, this is an important diagram because it shows you the ependymal layer. It will show you some of the astrocyte processes. You will see this is the brain tissue itself. And by the way, this is depicting the blood-brain barrier. I don't want to go into that. I want to concentrate on the blood, on the secretion and circulation of, Now, these are the brain vesicles, you know, that now, there. We have the cell there. Okay, this is also related, sorry, this is related to the blood-brain barrier. I don't want to go into that. I said I'm going to skip all of that. Okay, this is what I wanted to talk about, which is the projection of the arachnoid granulation. So the arachnoid meta projects through the wall of the dura, through the wall of the sinus into the sinus, giving us these projections known as the arachnoid villi, and the collection are the arachnoid granulations. This is an enlargement to show you how they form. These are the cells of the arachnoid meta, arachnoid meta, or the arachnoid, making this projection into the dura. These are endothelial linings. Because they are blood vessels, these sinuses are blood vessels, they are lined with endothelium, and the projection is through the endothelium, and the cerebrospinal fluid actually percolates. It percolates between the spaces, through the spaces between the arachnoid cells, making the granulations. This is another illustration, a side view. You can see these are the cells, arachnoid cells, and these are the epithelial cells of the venous sinus. And there is an opening there through which the CSF passes through. Here, I brought this one because it shows that the arachnoid cells are made both of pia and a few of other types of glial cells. These are the glial cells inside the arachnoid, subarachnoid space, and these are the cells of the lining of the arachnoid meta. And so through the spaces, the circulation takes place, or the movement of the blood through these arachnoid villi and arachnoid granulation. This is what I'm talking about. This is a superior sagittal sinus. It has got lateral lacunae. It's called the lateral lacuna. It's an extension of the superior sagittal sinus. So you will see the arachnoid granulations even when you look at the external surface of the arachnoid through the dura. If you look through the dura, you can see the arachnoid granulations, the arachnoid villi. So these are the arachnoid granulations villi found inside the superior sagittal sinus. So the fluid comes through these granulations and villi. This is another view with the superior sagittal sinus open, and you can very clearly see these arachnoid villi forming these granulations, both in the sinus itself and in its extensions, which are the lateral lacunae, So the cerebrospinal fluid is extracted from the blood, and then it comes back to the blood, extracted from the blood of the choroid plexus, and then it comes back to the blood to the superior sagittal sinus through the venous drainage of the brain back to the heart. When you remove the dura and look at the surface of the brain, you can very clearly see the arachnoid granulations. But this picture will show you that the arachnoid granulations are mainly concentrated around, inside and around the superior sagittal sinus of the dura and the lacunae around it. Now, one important issue, the CSF is found around in the subarachnoid space of the brain, but also around the spinal cord. If you need to take a sample of that fluid, you take it between the fourth and fifth, or the third and fourth lumbar vertebrae posteriorly. People take a sample from there, but there are precautions, infection precautions. Also, if it's a subarachnoid space, you don't do it because you will do sudden decompression, and the sudden decompression might lead to problems. But it's an important piece of information related to Csf circulation. Now, this is a summary. Lateral ventricles, we have the fluid produced there, also through the extension in the third ventricle. Then it goes through the aqueduct of Sylvius, more added in the fourth ventricle. We have a central opening, the foramen of Magendie, two lateral openings, the foramen of Luschka, and from there it circulates around the brain. Some of it goes down around the spinal cord. Now the pressure buildup will push it through the arachnoid villi and granulations into the venous sinuses and the lateral lacunae, especially the superior sagittal sinus there. This is the superior sagittal sinus. So the fluid goes in there and returns to circulation to the blood. This is a lateral view showing you the arachnoid granulation there. And this is the superior sagittal sinus in the junction of this of the far cerebri. But this is the subarachnoid space in which we find the fluid itself. That's it. Now, this is related to other dural sinuses, and I don't want to speak about that. So this ends this session about an introduction to the ventricles and the CSF circulation. Why I'm calling it an introduction? This is just an overview. During the practical sessions, during physiology lectures, you will reinforce this information about the ventricles. But if you still need more details about the ventricles, we can reschedule another lecture about ventricles and CSF. But to me, this outline, when it is complemented with the practical session and the lecture, physiology lecture, the picture will be completed in synergy. Any questions, any burning questions? You have 5 minutes to ask if you have any questions. Can you finish the attendance, please? Because I can see only about 12 attendants, but 33 participants, 33 attendees. Okay. I have good news for you. I found some backup copies of the lectures that I lost in the flash. I brought them along. I have a session now. When I finish, I will upload all the previous lectures and the recordings and everything to the site. This is one. Number two, Amina told me about your request to prepare a handout. I will do. The third thing she told me about flipping. Flipping means I'm going to post the PowerPoint presentation and some reading material before lectures. Then you can have a look at them. You can print them out if you are a physical person. Then you can write on the paper and all of that. No problem. I will do that. So 3 things. And thank you very much. I will see you later.